Hey, good morning, good afternoon, everybody. Thanks a lot for joining us. So we are kicking off today like a new webcast series. Our objective here is to speak to people and to teams and organizations who are like at the forefront of innovation in terms of leveraging AI in real life use cases to help us understand how AI is really like changing the world economy. And so today I'm very excited, very pleased to welcome Mike Nally. Thanks for joining us. Mike is a CEO of Generate, and Generate is a company based in Cambridge, Massachusetts, sorry, who specializes in using AI to design, optimize, refine proteins for therapeutics application, with a focus on immunology, oncology, and infectious disease. So the way we're going to use the next hour or so is Mike is going to give us like a short presentation to give us a context and give us like a quick teaching on what Generate is doing. And then after that, Mike and I will have a conversation, and we're going to talk about the business and how AI plays in Generate's model. At any point in time, if you want to ask a question, just write it down in your Q&A box or send it to me by email on pierre@newstreetresearch.com. And I'll do my best to take into account your questions and insert them into the flow of the conversation. And yes, with that, Mike, thanks again for joining us, and the floor is yours.
Thank you, Pierre. It's a pleasure to be here, and excited to share a little bit about what I believe will be a transformative technology for the future of life sciences. Let me just grab the slides here for 1 second. So Generate's a company that was founded now 6 years ago with a simple concept in mind. What if we knew how the underlying code of life in the form of DNA dictates protein function? And as all of you know, proteins are the most amazing machines, sensors, devices in the world. They program just about everything in biology. And yet the underlying code to function relationship had never been properly understood. And if you think about the overarching arc of drug discovery, historically, we've largely borrowed from nature to find the therapeutic advances that have treated society. So we have borrowed from, tree barks to create things like aspirin, molds to create penicillin. We then went into an era of rational and synthetic design where we were manipulating chemical matter to try and fit a target. We're now approaching an age where, because of some extraordinary data sources, some extraordinary compute capabilities, we can finally start to understand how DNA dictates protein function. And in doing so, it's driving this era of molecular and AI-driven discovery. But where we think we're going is actually even more exciting, where we can program biology in very intentional ways to treat individual disease with very scalable compute algorithms as well as hardware. And we'll walk you through some of those concepts over the next few slides. As we've seen historically, industrial revolutions occur when you see these massive step changes in efficiency. Electricity drove the mass production revolution, where we saw the cost per unit of electricity decrease tremendously. Compute, obviously, the cost per transistor has seen a similar trend. In biology, we've been really fortunate to see two coinciding trends. The cost of DNA sequencing dropped substantially, as well as the cost in gene synthesis. And when you couple this now with the cost of compute and the advanced machine learning techniques, it opens up biology to a wave of potential disruption that has been previously unimaginable. And it's very clear, if you look at the statistics, that biology has not been disrupted by the change in compute to date. So despite the fact that there's been a 25 billion-fold reduction in the cost of compute, the cost of every new drug approval is up about 28%, 28-fold over the same period of time. And so we're not translating these advances in compute to help us understand biology to date. And we think these new machine learning techniques will be at the forefront of doing just that. So at Generate, we've been actually pioneering two different fronts since the inception of the company, pushing cutting-edge machine learning algorithms to understand how DNA dictates protein function, but also inventing new hardware to drive an experimental learning loop to feed back and refine those computational approaches, because one of the limitations in biology is underlying data. This has required a whole host of different advancements, whether it be protein design models, proprietary data sources, new hardware, including miniaturization, microfluidic techniques, automation, in order to measure biology in distinctive ways, and then analyze that data to ultimately come up with new and better answers beyond what nature has discovered since inception. Where we think this takes us is ultimately leveraging a proprietary knowledge base with agentic approaches to do automated design and scaled experimentation, ultimately with a goal of designing precise medicines for precise patient populations. And we think that future is nearly upon us. So what is actually happening underneath the surface of all this? And while we're still a very young company, what we're seeing very clear signals of is that we're changing the underlying unit economics of drug discovery and development. Historically, it's cost about $2.4 billion of development costs for every new medicine. We're seeing about a 6% success rate. It's about almost a decade from idea to proof of concept, and about $400 million for each proof of concept. At Generate, we're seeing those numbers change in the very early data in very, very compelling ways. We've already seen 2 proof of concepts delivered. On average, those were about 4 to 5 years from idea to proof of concept. 80% of the projects we are working on have reached proof of concept, and what we're seeing is a dramatic cost reduction of about tenfold in the cost per proof of concept. And so the early signals are this will be a similar transformation that drives productivity in research in a way that we haven't seen to date. When we look across different the answers we're getting, what we're finding are functional answers that are somewhere between 20-fold and 500-fold better than current analogs. And these are in areas of huge unmet medical need, as you can see on the top of this slide, where you're seeing millions of people in the US and obviously global populations that are affected by these diseases. These sort of answers create more economical solutions for patients in an array of different disease areas. So where we think this is taking us and what's so exciting about the future is it's not just giving us better answers for existing medicines, it's also finding answers to problems that traditional techniques have not been able to solve. We have 2 tremendous partners with Amgen and Novartis. Independently, they've given us challenges that they've been working on somewhere between 8 and 12 years. They've not found lead candidates to those sort of challenges. In the first instance, we were able to solve and hit a lead within 2 years. In the second instance now, that cycle time is down to 3 months. And so again, what we're seeing is not just an ability to optimize the current state of medicine, but actually find answers to biology that was previously we were unable to interrogate. This leads to a pretty broad early pipeline for the company. The company is currently working on about 20 different medicines. Half of those are partnered between Amgen, Novartis, MD Anderson Cancer Center, and Roswell Park Cancer Center. The other half are proprietary assets, and we try and blend those different models of partnership and proprietary assets given the capital intensity of the work, but also the scalability of the underlying technology. And all of this is only possible through finding a great team. And I'm fortunate to work with some of the best technologists, best drug discoverers, best drug developers in the world that are passionate about solving the 2 fundamental challenges that plague the industry. One is this question of R&D productivity. We've seen R&D productivity go down for 4 decades in a row. And then, coincidentally, but somewhat interrelated, we also have inadequate patient access to medicines. And we think if we're able to solve the underlying productivity challenge for the industry, we can rethink the economic model to ensure patients get better medicines faster, and ultimately we restore, some of the image-related considerations to the pharmaceutical industry. Similarly, we have a great board of directors who have helped build other transcendent companies, and they've helped guide us in this journey. And so with that, I'll pause and, Pierre, maybe we can go to some of the questions.
Excellent. Thanks, Mike. I like it. It's very, very dense. Most of our audience are people more like looking at technologies and life science. So probably some of my questions will be really like focused on helping, clarifying the lay of the land and better understanding what you do. But of course, I think it's fascinating to see how is it like this combination of like very, very fast cost decline that you highlighted, like compute and gene sequencing and gene synthesis. This is absolutely fascinating. So maybe my first question would be looking back to your slide 6. So you mentioned algorithms, hardware. I'm pretty sure we are not talking about exactly the same kind of hardware. You're not designing your own chips, but like your own like life science tools, I guess. And so maybe could you help us understand how you integrate AI,
Yeah.
machine learning, into a physical process? Because you're not only designing like virtual molecules, you are actually making them and observing them. And of course, lead us into how through your chain you're creating data and reinforcing the overall system.
Yeah, it's a great question. And I think, Pierre, I think at its core, right, biology is the original information technology. And
Yes.
yet, up until 1953, with Watson and Crick, in the discovery of DNA, right, in many ways, we never knew how the 4-letter code of DNA drove all biological instruction. And that code has escaped human imagination for all of our existence. Despite now being able to stare at the code through companies like Illumina, which have sequenced now all species on the planet, we still have not been able to make sense of that code. Recently, we saw tools like AlphaFold, I think, take us from the ability to see the code, which Illumina kind of elucidated. What AlphaFold allowed us to do was read the code for the first time and predict folding patterns of proteins based on that code. What Generate has been basically trying to do since its inception is write entirely new text in that code to solve the greatest areas of disease. And generative models allow us to understand and learn from nature and understand the underlying principles in a way that have escaped human imagination and compose these entirely new texts. Now, what's important in that is that one of the rate limiters in biology has been our ability to see and effectively measure biology at scale. And if you think about how the industry in the biotech and pharmaceutical industry has historically operated, it's actually been on a reductionist principle, where we actually are trying to narrow down to an individual molecule as quickly as possible because of the cost of experimentation. And through automation, through miniaturization, through using these devices like microfluidics, which we call the hardware of experimentation, what we're seeing is an ability to radically reduce the cost of experimentation, increase the speed of data generation, to a point where when we started the company 6 years ago, when we looked at an individual target, we could only build about 100 variants to an individual target. And that would take a few months to design, build, and test those variants. Probably about, somewhere between 4 and 6 months. Today, we have systems that allow us to design almost infinite combinations. What's important, I think, to recognize is the size of protein space is unimaginably large. If you think about the average protein being 200 amino acids with 20 natural selections, the combinatorial possibilities are 20 to the 200th power, which is atoms on the universe cubed. And one of the things that, one of our board members, the Nobel laureate Francis Arnold, shared with me when I joined Generate was, Mike, you have to recognize that despite the majesty of nature, it's only sampled one drop of water in all the Earth's oceans of potential proteins. And so our ability to now start to sample at large is hugely, hugely important. And so today, with the cutting edge technologies, we can design millions of proteins. We can build a million at a time, and we can analyze them is in as short of a cycle as 8 days is our fastest cycle. And so the velocity of data generation on both biophysical and functional data is exploding to help us understand this space in a way that we've just never been able to understand it.
Okay. So let me rephrase that in a specific way I like to think about it. So you have a generative model that's looking at nature, at all possible protein designs, and you have an idea in mind, you feed that into a generative model that generates samples for you, and you actually physically manufacture these samples and learn about them and feed back your
That's
generative model to refine your understanding of the corner of the space you're looking at.
that's
Is that
exactly right. So historically, the way we would try and do this is we would have a biological target in mind. And we basically would immunize a human, a mouse, or a llama. We'd figure out what the immune system would, create a protein to attach to that target. And then we try and manipulate it to make it have drug-like properties. You got it spot on. What these technologies allow you to do is rather than going through that immunization campaign, if you understand the target you're trying to design, these generative models will design in silico DNA sequences. Those DNA sequences then are built into the full-length protein format of choice and then tested to see how well they accomplish the functional aim that you're trying to drive for that individual target. And your ability to do this now at a pace and a scale that, historically it's been about 3 to 5 years to get to from, kind of target to, initial human testing. We're seeing the time from target to drug candidate down to 3 to 6 months routinely, which is usually a 2 to 3 year period. And then the scale up is equally accelerated because of the quality of the manufacturability and developability criteria that you can co-optimize using these sort of machines versus sequential process in historical drug discovery techniques.
yes? So you're actually leading exactly to the one of the two follow-up questions I had. So the first one is just a clarification. When you said you moved from being able to do a few hundred in a few months to millions or almost unlimited in just a few weeks, all that you're talking about actually producing physically,
Yeah.
right? And so that's more like the gene sequencing and gene synthesis improvements that you showed on your side, and then that feeds back into the machine learning.
That's right. And then we're pushing even cutting at further techniques, like rather than use cells to grow proteins, could we actually use cell-free expression to cut that time down? So everything we can do, there's an obsession within the company for speed and scale.
Okay.
As you know, feeding these machine learning models is all about, the velocity of data generation to ultimately refine the underlying answers that you're getting.
Yes, exactly. Um, so maybe tell us about your generative models. Um, what's the baseline? I guess you fed them, like in terms of the data you use to make these models to train or
Yeah.
fine tune. enhance the models. I imagine the initial data set is more or less nature. So you feed it with all the information you have on nature, and then you feed it with your own data that you're generating through your processes. Is that right?
That's exactly right. So there were what made proteins a tractable problem. And this is why the Nobel Prize was awarded to AlphaFold. There were two foundational data sets that were publicly available. One was the sequencing revolution, sequence every species on the planet. That gave you 180 million amino acid sequences across all species. The second thing, and equally important, was there was this obscure structural biology database called the Protein Data Bank that was an exceptionally well curated database of protein structures. So the structural biology community used to solve these artisanal structures of individual proteins. They had deposited over the last 40 years about 200,000 different representations. And while that 200,000 points doesn't seem like a lot, these are extraordinarily data-rich sources. So every structure contains tens of thousands of data points because you're actually looking at the 3D coordinates of the structure. And so by understanding those two sources, you had the ability to discern sequence to structure interrelationships. But if you're making medicines, sequence and structure are nice intermediates. What you care about is function. And so generates hardware that I talked about earlier was largely established to create a functional understanding of protein space so that you can now add the third leg to the stool. So I understand now sequence, I understand structure, but importantly, what I really care about is how that protein will function in a human system.
Yes. And so what's your so maybe we'll come back to that later. Okay, so give us a sense. So how big are your generative models? So everybody knows about like the Rageous scale of like ChatGPT and Grok and others. Are we talking similar size? Because your universe is way, way, way richer than
Yeah.
human language, of course, as you say, I like the number of atoms in the universe to the cube. So does that mean your model are even more gigantic, or are you working with smaller models? How does that work exactly?
These actually are smaller models than large language models. And so, and this is the conundrum with biology. Biology has not been measured as often in reproducible data formats as obviously we've created in language, right? So the number of data points in language, when you can scour the internet, is far greater than the number of data points you have in biology to date. We think with these sort of tools and with better measurement tools, that will start to change. We're training our models. We use a cloud provider, Lambda, to train our models. We use about 256 GPUs in the training step. Obviously, that's a small fraction compared to what the most cutting edge large language. I think the largest biology model out there is somewhere between, probably about 1,000 GPUs for a couple of months. And again, it's not nearly as data rich a regime. We actually are probably seeing greater benefits in actually the post-training steps than the training steps right now. But the just to give you a sense of the pace of change, we quadrupled our compute last year. And my guess is it's fair to say we'll continue to see a similar trend in
Yes.
that direction.
Okay. So your pre-training is that kind of scale, very decent, like hundreds of GPUs. It's already actually
Yep.
gigantic models, but it's not like hundreds of thousands of GPUs. And then I imagine um, like your post-training activity must be very, very intense if you're generating like millions of data points, like in note, like you mentioned, you said unlimited. So you're
Yeah.
generating So there is a kind of like a permanent process of refining the models. And is that something you would qualify as post-training? It's not pre-training, like the pre-training.
That's exactly right. Yeah, and so and I think your ability to tailor, we see just huge advances in the ability to tailor those models.
Yes.
And so, I would guess today, if you were to think about pre, post, inference, it's probably about a third, a third, a third. I think in the future, we'll probably skew more to post training and inference. We'll
Yeah.
continue.
Yes, exactly. Because so okay, let me um, shift shift gears from here and go to the growth model. So maybe I got your numbers wrong. They're so big. So it used to cost like billions or hundreds of millions of dollars to get to proof of concept. Now you're getting to 40 millions.
Mhm.
So it's a 10x reduction. And given your underlying model, you should get to 4 million. I don't know if it's in 2 years, like or in 10 years, but you should get another 10x uh,
Yeah.
over time. So the cost of this proof of concept is going down to the ground. Um, and of course, a lot of people think that as being like deflationary, meaning like, things are getting cheaper, so you will need to spend less money. But like, excuse my lack of understanding of life science, but like the number of proof of concept you would want to do is almost unlimited because you could imagine doing a proof of concept for like any individual, any disease, any specific problem. So can you help us understand how
Yeah.
by, let's say you take the cost of a proof of concept down by 100x,
Yeah.
how do you expand the addressable market of proofs of concepts as a whole, basically?
Yeah. So I think I think, I think we need to unpack that cost curve a little bit because I think what these technologies today are doing is they are the cost to getting an optimized molecule to pursue a lead is will commoditize down to zero. And to me, that is extraordinarily powerful. We used to think that was about a $50 million expenditure when I was within the large pharma universe. You're going to be able to basically print molecules to prosecute the biological hypothesis that you're trying to pursue. And we can debate the time horizon that happens over. My guess is that it's within like a 3 to 7 year window. That cost curve almost comes down to zero. Right now, even where it is, we've already seen the biggest deflationary pull down, where now we're in the hundreds of thousands of dollars versus the 50 million. Um, where cost accrues now is more in the manufacturing scale up, the animal models, and the early clinical trials. And so I think the inevitable next step for computationally native biotech companies like ours is to start to use these techniques to similarly drive efficiency in those other domains. So the FDA just released guidance recently on a desire to move away from animal testing. The reason they want to move away from animal testing is animal biology is historically poor at predicting human biology. But as you can appreciate, for ethical considerations, we wanted to make sure that we tested things in other species before we put them into humans. With better predictive models, with new experimental capabilities like organoid systems, which basically are synthetic organs, we can now have much more predictive preclinical systems that will have a similar deflationary drive to cost. Clinical development is then the kind of last frontier, and it's actually where the majority of cost in the drug discovery and development process sits. It's where the majority of time sits. But if you think about like this natural progression almost at a unit level, and this goes to the complexity of biology a bit, proteins are kind of like the base unit. Then you go to cells. Then you go to tissues. Then you go to organs. And then you go to organisms. being human beings. Like our ability to master that continuum is still a ways away. Now, what's becoming clearer is we are starting to decode biology in ways that we are understanding the language of proteins today. Cells will be the next frontier, and it will continually scale up, even though there's not pure linearity across each of those systems. There are some emergent behaviors that you start to see, especially given the complexity at different levels in that cascade. And so for us, part of what's so exciting is we believe this era of programmable biology is starting to take hold. It's a long road to fully master the complexity of the human system and be perfectly predictive of it. We need a lot more measurement. We need a lot more clinical data. We need a lot more of the type wearable type data that we're all starting to generate. But we're on a road to making biology engineerable. And if you think about like the mass transition that we're really talking about, it's actually taking what has been an artisanal endeavor of drug discovery and making it engineerable. And that brings with it all of the predictive qualities that we see in other engineering disciplines, as well as efficiency qualities. And I think that's what gives us so much hope that we're on this beautiful arc to actually understanding human biology and solving it in really new and distinct ways.
This is fascinating. So let me rephrase that just to make sure I get that right. So today you have like your three scaling laws that are like vertiginous, and they're all about figuring out a protein, manufacturing it, and testing it.
Yep.
And here you're going down, like you went from 50 million to a few hundred thousand, and you're going down to zero over
Yep.
like a few years.
Yep.
But then what you're saying is that after that, the next steps are like manufacturing and trials, or is that trials and manufacturing maybe?
Well,
Or
I would actually take, so what we ask ourselves is the first order question is when you have basically printable medicines,
Mhm.
molecules on demand, what will that allow you to do differently? And
Yes.
one of the things it'll allow you to do differently is it'll allow you to interrogate biology. And this is I actually think it'll reform and revolutionize hypothesis generation first. So the way we generate hypotheses today to biology is we do it on an individualized basis. We sit there and say, all right, we understand this pathway in the immune system, and I want to go after this individual target because we don't have the capacity to interrogate all targets at the same time. So we have to kind of take a reductionist approach to hypothesis generation, isolate the individual target, find a molecule, manipulate that molecule to then study it ultimately, in preclinical models and then human models. Where I think this is going very, very quickly is our ability to not presuppose the hypothesis. So what if I'm trying to kill a cancer cell? And rather
Yes.
than saying I want to go after a very specific target on the cancer cell or a specific mutation, what if I say, well, now that I have the ability to print molecules on demand, let's look at every receptor on that cancer cell and see which one kills cells best. Let's understand the combinations. I don't need to presuppose the answer if I have the right assay to read out the ability to kill cells. And so I think this is going to allow us to interrogate biology differently in the first order. But you're exactly right, is the next order steps are going to inevitably say, okay, well, now how do I find better preclinical systems that are predictive of human biology? And then how do I actually predict human biology? Because you could imagine in the future having a digital twin and intervening at a precise location for your individual condition and being able to see what is the biological readout. The more predictive that becomes, the better we'll be at it. And this is, I think this is very much akin to, very complex systems like predicting weather, right? If you think about, how scientists have tried to solve these things historically, we've taken bottoms-up approaches but the complexity of the systems actually overwhelm, where small perturbations lead to dramatically different outcomes. These machine learning models are picking up patterns that are indistinguishable to our human eye, but are allowing us to understand these very complicated systems in a way that we've never been able to do bottoms up.
So if I understand you correctly, ultimately, in like the personalized medicine kind of ultimate vision, which is I am diagnosed with a cancer, and like there is like a workflow that can produce a molecule for me and that is going to treat my cancer. Machine learning is not just going to be about figuring out the right protein. It's going to be, I mean, it's going to be figuring out the right protein, but down the whole chain, including testing and
Totally.
um, like what is today like the clinical trials will become like a sort of a trial just for my specific uh,
Yeah.
situation.
Yeah, we'd love to get, I mean, like that is the distant dream. I mean, the distant dream that we have at Generate. And we sometimes take some flack because, it is extraordinarily ambitious. But it would, where you ultimately want to get to for society is to be able to instantaneously generate the optimal therapeutic for each individual's condition.
Yeah.
Like that is nirvana to us.
Mhm.
Um, I don't think we're going to get there in, the next 5 years, the next 10 years. I think there's huge regulatory changes that would have to occur. But I think we're on a path toward engineerability that's indisputable. And if you just think about the complexity, one thing that I think is very fair to say, if you think about the advances we've seen in medicine over the last 100 years, so you think about all the new vaccines that have saved lives, you think about things like penicillin, antibiotics, infectious disease meds, cardiovascular meds, um, cancer meds that have, made a significant difference in life expectancy. We still only understand 5 to 10% of biology, despite those monumental advances. So just almost play a conservative scenario. Over the next decade, I think it's very easily possible, highly likely, that we will double our understanding of biology, maybe triple it. So if you think about in the next decade being able to have at least the same advancements we've had over the last century in medicine, or maybe twice as much, the amount of human suffering and the benefit for humanity will be extraordinary.
And it is a very, I don't want to say linear, but continuous kind of journey. You don't, one concern would be, okay, I'm able to generate these millions of proteins very, very rapidly, instantly, but then I still need to go through clinical trials, and it's a real mess because I
Yeah,
mess in terms of
totally.
it's like it's going to be 10,000 times slower. So there would be a bottleneck. But the reality, I guess, is that as you're improving the efficiency of the discovery part, then like I imagine like the rest of the chain is going to actually progressively become more efficient. So you just mentioned like moving away from animal trials and
Yeah.
doing more synthetic trials. Can you go through that again and give us a sense of
Yeah,
what's the
totally.
time scale for that and how is it going to increase the efficiency of the step right after you have a good proof of concept, basically?
So maybe I'll take the easiest stuff first and then go to the harder stuff. So the easier stuff, right now from the day a trial concludes to filing is usually a 3 to 6 month process. So this is study closeout, data scrubbing, report writing for regulatory submission. And if you attach an LLM to the back of your data feeds, those reports can be drafted almost instantaneously. So there's 3 to 6 months of efficiency right there. And if you think about, the average drug, every day is about $1 billion of value that you're saving. If you think about, time value of money of peak sales of $1 billion product a year, that kind of gets you there. So that's like the lowest of low-hanging fruit. Now, think about every patient who's diagnosed with a new disease. So, the first thing they do is they Google. There's now a digital identifier on every patient out there. So what's the other thing we can do? We can change recruitment rates using advanced computational approaches to identify and enroll trials with the right patients for these medicines in a much more timely fashion. The piece that I think is going to be harder is how do we become more predictive of biology to comfort regulators that we're thoroughly studying these medicines. And I think we have to tread somewhat carefully here, where we don't want these AI designed medicines to have a different standard than we've historically applied. At the same time, are we able to enrich patient populations knowing underlying biological signatures to get to bigger treatment effect sizes that allow you to create smaller trials because your statistical sample needs to be smaller? Those are all things that are within our reach today. Over time, I think, what we're going to try and do, and what we think is a tractable problem again, is as we start to understand the complexity of each of these systems, so going from the protein to the cell to the tissue to the organ to the organism, as we start to be able to effectively model those things better, ultimately there should be compelling arguments to regulators that why are we running trials when we actually can simulate these things with a high degree of confidence? That's a longer way out. I don't think that's a tractable problem in the next decade, but I think, we're on a path to that sort of disruption.
Okay, it's interesting, and it's actually a very generic um, situation or like roadblock to AI. You have the same like for autonomous driving, for instance, which is
Exactly.
if you really want to get autonomous driving at scale, you need to have cars without a wheel, without someone at the wheel driving people around. And if you do that, like the economic value creation, like the improvement of life quality of everybody is insane. But at some point, you have to convince, in some ways, the regulator that it is safe to do so. And it's a very interesting because it's a complete change, almost like deontological change in the way you consider risk. So today, I guess the regulator is convinced that doing a trial is a way you can make sure like a new protein is safe. But that's like an accepted
Yeah.
commonplace that has very very little, ontological substance. So we just need to move to a place where people are like, no, if the model said it's safe, that means it's safe, and you still have like a very small risk. And I guess what you see in autonomous driving is that the regulatory problem looks like an unbreakable barrier up to the point you start taking off very slightly, and then suddenly it goes very, very, very fast because regulators adapt actually. They kind of like need like a rear mirror view, but once they have it, they adapt like actually relatively fast.
I think that's spot on. But I think what's exciting on the journey that we're on, whereas like in autonomous driving, until you crack that regulatory barrier, you don't unlock the market. There's huge unlocks all along this journey for us. Like just getting to the
Yes,
lead faster already is much better than what the traditional model would suggest.
yes.
The thing that one of the things we haven't talked about a lot is these tools will allow you to drug the undruggable. So
Yes, exactly.
right now, if you don't have an ability to prosecute a target, then you don't ever have to go through that process. But now we're going to have tools that will allow us to perhaps address something like Alzheimer's disease. Those things create huge pockets of value as you kind of traverse this process toward that future state you just outlined.
Yes, exactly. And isn't there also like an element of, in my understanding of how things work today, you have like a protein candidate or like a cure candidate, and then you recruit patient to test it. And these patients you recruit are people who are very ill, and they see like being recruited for a test like as a blessing. And this is this idea of testing a medication that will be used then for many, many other people. But if you get closer and closer to a medication which is for a very small population, a very rare disease, then suddenly like your test becomes your cure, more or less, no? Or isn't
That's
there
right.
something like a
Without a doubt.
place where actually things unlock by themselves? Or
There's no doubt about it. I mean, like, and that's what all of this just harkens back to the fact that we just don't understand biology very well. And the models that we have today are not very predictive, right? Like if we've cured cancer in mice countless times, but the reality is the mouse model doesn't translate to the human model. And therefore we don't have the same things. What these tools are allowing us to do is have much more predictive capability on what
yeah,
works, in what diseases, and at what points in time in someone's life. And again, you don't need to solve it all. It doesn't need to be a binary moment where we go from no advance to huge advance. This is a continuum that
yes,
we think is accelerating in pace given the technological advances we're seeing.
yes, that's very clear. Um, okay, so maybe tell us about the journey from here. So you said you're spending about a third of your compute on pre-training, a third on post-training, and a third on inference. Um, and how this proportion will grow, I think, is anyway anyone's guess. It's difficult to know. Everybody has been expecting inference to grow much faster, and there are periods it's growing much faster, and other periods you have innovation in training and post-training that make it grow even faster and catch up. So, uh, but my question would be more, how much of your total cost is compute today, and how much is it going to be in 10 years? That's the
It's
real question.
Yeah. So right now, I mean, and this is just, again, the nature of the model. Clinical trials is your most expensive piece
Yes.
by far. So our compute costs are about maybe 8 to 10% of our budget. We think that will grow at a disproportionate rate relative to headcount costs or clinical trial costs. But at the same time, um, it's still a small fraction of our overarching expenditure. And so, as far as I can see right now, um, I think, uh, while I think it's fair to say we'll probably quadruple again over the next 12 months. Obviously, we're getting some performance benefits as well. We will be very compute hungry. I think the shift will be still more to the post-training and inference. So I think those will skew higher, if I were just to guess. But as you know, sometimes you run these experiments and you see remarkable gains in a domain. This equation is a moving target to a certain extent. As you add different data, as you add new models, you're trying to model different systems. So you can imagine today we're modeling proteins. Tomorrow we may be modeling cells, and therefore we're going back to new training regimes.
Yes, exactly. I guess for now, rephrasing what you said just to make sure I get it right is you train on nature
Yep.
and then you post-train tailoring to a specific situation, like a very, very narrow part of the gigantic space you're searching. And so as you grow your business, nature is not growing. So like the training is not growing as fast as post-training or inference.
Well, so when maybe slightly rephrasing. When we started the company, the ratio of public to private data was 1 to 0, obviously.
Yes.
Today, the preponderance of our data that we train on is proprietary data. So the number of data
Right.
points so
It
every
went from 1 to 0
protein,
to
it's probably about 1 to 2 right now.
1 to 2. Okay. Uh,
Um,
and
and
it's going
that
to
and
keep
that's on
going
a
that
massive
direction.
increase, right?
Yeah.
So we learn on every protein. So our models get better with every protein we generate.
Okay. Um, so a couple questions on that one. So you're working on a specific cure or on a specific line item in your pipeline. Are you generating their data that is going to be useful to the other lines of your pipeline? And is that like a no-brainer? Like there is a very significant overlap?
Yeah, so different data types some data types are very generalizable, some are less generalizable. Functional data is usually specific to the individual therapeutic target, whereas biophysical data generalizes quite well. So biophysical data is stuff like binding affinity, like understanding the
Okay.
interactions.
Yeah.
When
So
we
which is
start to
more like
understand one, exactly, that generalizes quite well. Developability data. So how developable or how manufacturable a protein is. What is the viscosity of a protein? The immunogenicity data. All of those types of data generalize broadly. Functional data is much more specific because if I'm trying to modulate a cancer target, I'm trying to kill the cell. If I'm trying to modulate an immune target, I'm usually trying to modify a certain signal.
okay. Okay, excellent. So that's a awesome transition to my next question, which is again, I know nothing about life science, but I've heard this debate about you have the sequence, you have the protein, you have the structure, and you have the function. And some people say, well, look at the structure, it's very important, and you have AlphaFold, etc. And others say, no, 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 you want to relate directly the sequence to the function in the way you're creating your connections and building your model. But so what you're telling me is that things that are more like alike structure, which has intermediary layers in the structure of biology of life, are actually places where you find the most generalization. So, and that's basically very high value, right?
That's right. And we made our biggest single capital investment since founding the company 6 years ago was actually a structural generation capability. So
Okay.
we bought 4 cutting-edge cryo-electron microscopes. I mentioned this protein data bank earlier with 200,000 structures of proteins. Interestingly, the vast majority of those are freestanding proteins. When you're making therapeutics, what you care about is interactions. So what I want to actually see is the antibody antigen interaction because that's what's modulating the biology. There are only 2,000 of those examples in the public domain. So we built a core to make cryo-EM antibody antigen structure determination at scale. We will have more data than the public domain in the not too distant future. We're solving thousands of structures a year at this point. And so we've seen this data loop, which allows us to model interfaces better than anyone else on the planet.
Okay. So one of my questions was, do we care about intermediary information, or do we just care about sequence and function? And the answer is absolutely like there is no question. It's very, very important to understand intermediary
Well,
information
it provides generalizable
because that's
features
where.
that allow us to determine function better.
Okay, great. Um, so you guys at Generate are doing that. You're generating your own data. You've already tripled your data set from what you got from the public domain, and it's going to only accelerate. And I guess a lot of other people are doing that as well. And so are you guys looking at each other and being like, what kind of data do you have? What kind of data do I have? And do you think there will be like a market for that data and that you're going to start like striking deals to
You know,
or do you think it's going to drive consolidation like that to generate merging with another business and because of the data would be a no-brainer in a few years?
it could. I mean, I think, part of the reason we have a deal with MD Anderson Cancer Center MD Anderson Cancer Center collects 2 gigabytes of data on every cancer patient that walks through their doors. They joke that the secrets of cancer sit in their freezers because of all the biopsy samples they have.
Mm-hmm.
Right? Like that's a data set I could never recreate. What they do is they look through that data, they see identifiable markers that represent the next generation of targets. They share that data with us, and then we design the molecule, and we do 50-50. So partnerships is one way we really lean
Yeah.
into to get the best of our capabilities, the best of someone else's capabilities. I was at a breakfast this morning with one of the other leading AI ML drug discovery companies. They focus on a completely different task, more in the target discovery space. I could imagine a world where there's huge synergy between the work they do and the work we do.
Mm-hmm.
Those would be, whether it's a partnership, whether it's, acquisitions, inevitably these capabilities need to mesh in order to treat as much disease as fast as possible.
That's very clear. And it's fascinating. So your machine learning, your AI is heavily integrated into a very physical, very real world process. And actually you've innovated a lot to generate a lot of data, and it's a self-feeding system, and the data is inherently proprietary. Basically,
Yep.
you generate it, and there is no shortcut to get it elsewhere. Or
Well, one of the funny things about it, Pierre, is actually, when in the early days of generative AI, people would focus it disproportionately on hallucinations.
yes,
When you build every protein that you're generating sequences for, hallucinations become features, not bugs in your process. Because we learn as much about the hallucinated sequences that don't function as we do about the functional sequences that function. It tells us
because you
something,
manufacture all of them. Yes. Mm.
and we refine, then all that data is fed back to refine the computational approach. So, what we're doing for the first time in molecule discoveries, we're learning from everything we work on. And it's giving us an ability to refine at this extraordinary rate.
Yes, this is great. Let me check. We got a lot of questions while we were chatting, and so one question was, has Generate shown a reduction in clinical failure rates with AI ML? So I guess you answered that. You said yes. That's
Well, we're starting to, right? I don't want to declare victory too early. We as a 6-year-old company, from concept to finish of an average molecule in our industry is 15 years. We think our first molecule will get to that finish line in about 8 years. So it's too early to say, we have categorically cracked that equation. All of the interim markers are suggestive that we will see a differential success rate. I mean, the average success rate from a molecule from concept to fruition has been 5 to 10%. And we think this will have a material positive impact on that number.
excellent. And I think we've covered all the other questions, and we're on the top of the hour. So Mike, thanks a lot for taking an hour to share all that with us and to educate us. It's fascinating, and my team and myself, we are particularly excited to see like this convergence of technology and life science. It's really fascinating, and it's very encouraging. As you said, it can really have like a very incredible impact on positive impact on mankind. So it's really, really great. Thanks a lot for that.
Thank you, Pierre. Appreciate the opportunity.
Let's stay in touch and speak soon. Bye.